welcome back. The second talk of the day is Algal Biotechnology. I hope some of you are working on algae. So we're going to cover different species of algae. There's a lot of pictures, a lot of different names, families of algae, and then towards the end we see some application of using algae. Okay. So that's how algae look like. We have algae that's from microscopic to really big algae. Like in seaweed, algae can grow in really 10, 50 meters long. Really. So algae is a big biodiverse organisms. Okay, can be microscopic to really a big organism. So what is algae? So algae is a group of plants, a plant-like organisms. It's not actually plant, it's plant-like organism because it can do photosynthesis. It doesn't have root stems and leaves like plants, but it has chlorophyll and it can do photosynthesis. It has chlorophyll A as their primary photosynthetic pigment that are commonly present in freshwater and marine water algae. Okay, so that's basically what algae is. That they have chloroplasts and they can do chlorophyll with a pigment that can carry out photosynthesis by fixing sunlight. So most of the algae are eukaryotic. There is the misnomer about algae prokaryotic. It is not, because there are cyanobacteria. People call the algae. It's not algae. It's a bacteria. Okay, so that's why we put that. Is it prokaryote or eukaryote? But almost all algae, in my opinion, it's eukaryotic organism. The cyanobacteria people call algae, blue-green algae. They call it. It's not algae. It's a bacteria. Okay. So they are single-cell species, the microalgae. Okay, examples of prokaryotic microorganisms is cyanobacteria. We call this blue-green algae. They are photosynthetic cyanobacteria, responsible for creating the earth as a livable earth because of algae, photosynthetic algae, cyanobacteria. Because the earth was, one time it was completely unlivable. It was really hot and hostile place. It's an anaerobic condition, no oxygen at all. Because of algae, it created 21% oxygen in the atmosphere. That's because of algae makes this world livable. Okay, so we all have to give a great depth to the algae for making the earth livable. So the difference between prokaryote and eukaryote, because of this one algae is prokaryote, you need to know the difference. The prokaryote cells are simple cells. They don't have nuclear membrane. The DNA is right in the cytoplasm. The eukaryotic organism has DNA covered by nuclear membrane, and that's one major difference. The other difference is in prokaryotes you don't have cellular organelles. Like inside the cell there is subcellular organelles like endoplasmic reticulum, mitochondria. All that you don't have it in prokaryotes. In eukaryotes you have a lot of subcellular organelles. Okay, and those are some of the major difference between prokaryote and eukaryote. The microalgae are very small, measured in micrometer unit. There's a single chromosome present in prokaryote. There is no membrane-bound cellular organelles. In the eukaryote are basically exactly like you and I. We are all eukaryotes because if you look at your cell, except the chloroplast in the algae, everything is almost similar. In the cell wall, we don't have cell wall, but it has the subcellular unit exactly like us, mitochondria, Golgi complex, endoplasmic reticulum, all that thing in eukaryotic cell. So that's why almost all algae are eukaryotes. Then eukaryotes could be microscopic eukaryotic algae or macroscopic, very big ones. Okay. So microalgae examples are eukaryotic microalgae include chlorophyta and bacillerophyta. Those are some examples. These are photosynthetic microorganisms. They capture sunlight and fix carbon dioxide in the atmosphere, convert carbon dioxide to carbohydrate. Okay, so they make food by fixing carbon dioxide combined with water. So carbon dioxide is converted to carbohydrate. The electron is coming from hydrogen from water. So you use this algae also, when you grow in certain condition, can accumulate lipids, like the other day I talked about, when nitrogen limiting condition. When they make a lot of lipids, they float. I use a lipid oil to help float in the water. That's another purpose of it. It also produces a lot of pigment that could be used in So why they make pigment? They make pigment to absorb sunlight and also reduce stress. Okay, the biology doesn't get, it will get stressed at too much sunlight. That's the reason they get a lot of pigment. Every algae has a lot of different pigment, different colored pigment other than green. Okay, that's the reason for that. So microalgae contain different types of chlorophyll, which is all algae contains chlorophyll A. Chlorophyll in cyanobacteria is distributed throughout the cell, and you can see in this picture, that's how a chlorophyll look like. The whole cell is full of chlorophyll. Okay, so that's how a chlorophyll looks. It keeps switching off. Okay. Thank you. All right, back again. The whole cell is covered with these chloroplasts. You can see the structure of chloroplasts with thylakoid membrane, and this is where the sunlight is captured, and you get the electrons agitated. That helps the algae to make this energy molecule called ATP, and an electron transfer called NADP. Okay, that carries electron, it become NADPH molecule. So that is important, because in order to make those electrons agitated to make ATP and NADPH, you need light. That's the reason algae need light. Once you make those two molecules, then it's a dark reaction, where that two molecules is used to make your carbohydrate through Kelvin cycle, another pathway to make that. So microalgae contain chlorophyll within the membrane to form chloroplasts. Chloroplasts are cell organelles that capture light energy and convert into chemical energy. That's a reaction right there. Solar light is captured, water and carbon dioxide is combined, and you can make glucose. So what's happening is, here is the electron is taken out and put it in here. You make your glucose molecule. The end product is not only glucose, you produce oxygen, right? This is really good to keep the earth oxygenated aerobic conditions. Chloroplast has a double membrane like structure like nucleus. Sunlight is trapped and used for energy. Chloroplast contains this green pigment called chlorophyll. There are a lot of different type of chlorophyll. Different algae has these different type of chlorophyll. There is two pathways called electron capture, photoelectron system 1 and 2, and the major difference is the one is electron capture and use, 2 is electron is recycle, okay? Capture and then recycle back into the process. So it's an example of Euglena, one name of the algae. You see, look at it, 85% of the cell is chloroplast, all those green stuff, right? So if you look at that, all of them are lots and lots of chloroplast in them. The main thing is to capture the energy and make the food and release the oxygen. That's all it does, 24/7, non-stop. Even there is no sunlight, they make it because they capture the electron. At night time, they make the dark reactions. Okay. So, how a chloroplast look like? That's the structure of your chloroplast. It's a double membrane, and your electrons are transferred in between this membrane. And that's where we call that electron transport chain or system. Let me move it again. So this is a double layer structure. There are 2 layers. The electrons are transferred on this, in between this layer, and when the electron that carries electron and goes into this NADH, NAD become NADH and ATP is used, and this biochemical pathway, there are a lot of chemicals produced, okay? That cycle is called Calvin cycle, named after Benjamin Calvin, the guy who first figured this out. So that's the main machinery of chlorophyll, chloroplast, okay? Some of those pigmented algae has different color to it. Okay, that's pigmented algae, like brown, red, different color. Okay, so here's some of the examples. This is the algae called Porphyridium. Another one is Chlamydomonas, one of the common algae. We call green algae. We call this red algae. Okay. So here is a list of major algal group. Okay, this is the algal group. There are a lot of different names out there. And these are some of the common names that people commonly call that instead of the scientific name. And you can see the difference in chlorophylls. There, some of them are A, B, A, C, E, A, B, different type of chlorophyll they have. And then the major storage products in this algae are mostly starch. Some have lipids, some have polysaccharides, and some have oils and starch. So those are the the different product they make depending on what algae you are cultivating. Okay, there's some structural difference between these algae. Some have flagellum, so they use a flagellum to move around. Some have no flagellum. Some have multicellular. Some are single cellular, and there is a lot of different difference between these group in there. Okay. So another difference is these algae, some are marine, saltwater algae, some are freshwater algae. They like only freshwater. Okay, so those are the major groups listed here, right there. Some of the common names are blue-green algae, green algae, golden brown diatoms, euglenide, brown algae, dinoflagellate, red algae. So you can see that common name, scientific name next to it. So let's look at the differences. So the name Cyanophyta, actually a bacteria with photosynthesis characteristics. This is what we call blue-green algae, the cyanobacteria. Okay, it's a misnomer. It is not a algae; it's a bacteria, but people call it blue-green algae. And then we have Chlorophyta, is a green algae. It could be micro or macroalgae. Some are large, like your sea lettuce, we call vulva, scientifically vulva, and then we grow near coastline with high nutrients. Examples are chlorella, Sendamonas, Chlamydomonas. Those are all belong to this group called Chlorophyta algae. Okay. Then we have a group we call Chrysophyta. This is commonly we call diatoms. And these are most important among the Chrysophyta. They have 2 parts. They have the shell to it that fits together and is full of silica, impregnated with silica. That's important for in the seashore and other things. There's a lot of silica in it, and you have the dinoflagellate dominate this ecosystem. Then you have the Euglena phyta, we call that Euglena. It's a single cell algae, is a very, very common one. You can find this everywhere. Mostly it's in freshwater algae, found in freshwater. Then we have Rhodophyta, we commonly call red algae because it has a red pigment. It could be micro, it could be macro, bigger size, smaller size. Mostly marine, have high amounts of the chemical called phycobiloprotein that gives the red color to the algae. Okay. So some of the examples of this algae are Gracilaria, and we have Porphyridium. Those are all name of the algae that belongs to this group Rhodophyta, commonly known as a red algae. Okay. Then we have Phaeophyta, which is the brown algae, and color is brown because the brown pigment is no microalgae, it's a macroalgae, it's really big size, you can see with your naked eye without microscope. It's marine in nature. This includes the seaweed, or we call kelp, right? It grows in meters and meters of length and in marine system, which has this incredible growth rate. They grow really, really high. It's a highly productive algae, and we take this and we make a lot of things with that. You know, Japanese, they eat the main food source for them, rich in protein, and also this is we used in the microbiology lab to make agar. Make agar. That's the ingredient coming out of that. Okay. So this is some of the colored picture of the group called Cyanophyta. There's a lot of different shapes. There are spirillum, they are rod shaped, and you have circular shape, all kinds of different shape of algae out there. So here is some example of the spirulina plantis right there. So some of them are common in Hawaiian water, and this come from Hawaii culture collection. So Cyanophyta, actually bacteria with photosynthesis. Cyanobacteria is, as I said before, it includes spirulina, and anabina is another example that belong to that. This is another group. This is the Chlorophyta. That's how it looks like. This one is like a large one that can grow really big, and we commonly call this sea lettuce. And then we have the smaller one. Those are Chlorella, Chlamydomonas, Centomonas. They're all right here. Okay. So these are all belong to this group called Chlorophyta. It's commonly known as green algae. Which one? I don't know whether it will play here. Can you try that? Yeah, play that video. Sometimes it works. You can see that under the microscope. That's how they move around. So really active culture right there. Okay. So you can see that this whole group is Chlorophyta. Okay. Yeah. Thank you for pointing that out. Okay. Then we have Rhodophyta. Oh, let me go back. The Rhodophyta red algae is again macroalgae. It produces red pigment. Here is some of the example. The Gracilaria, two different species, and then Porphyridium, which is the single cell one, and then multicellular one. All of them are out there. So it's produced this red color. Okay. Then we have the Phaeophyta, which is brown algae, mostly macroalgae. Marine includes sea kelp. That's how it looks like pictorially. You can see them. They are really, really big algae grown in the sea. Okay, we call sea forest sometimes. We have Cyanophyta, and Cyanophyta looks like that. Example is the diatom. I said it is rich in silica, and it has the shell, and it also can carry out photosynthesis. If you want to play that, you can try a video again. No, it doesn't play much. Okay, that's how a cyanophyta look like. Okay. Come on. Can you change that? I can forward this. Okay. One more. Okay, got it. Then you got the euglenophyta. The euglena, which the common one, is a freshwater algae, and that's the euglena here. So euglena also can move. It has flagellum. So try that again. It will move. It will swim. Just to show how it looks like. No? There you go. That's how it looks like. Oh, yeah, somebody is pointing out just a small video we are showing. They're saying they cannot see that video, it looks like. Okay. All right, next, let me control here. Let me smooth this. Can you change it? I can move. All right. So, can you move this down? Yeah, yeah. So, the current commercial application of algae, so you can see a lot of product you can make, and depending on the species of algae, whether it's chlorella, whether it is Duvanella salina, you can make health food, beta carotene, some of those DHA, which is our important for cholesterol reduction, and we can also make aquaculture feed. These are the places you can commonly find these algae and are commonly used for a variety of sources. The main culture system is how they grow in large quantities, be a tubular bioreactor. They grow in really big culture, or they use a raceway pond system. So there is a lot of extensive open raceway pond, or photobioreactors, or sometimes it's a heterotrophic cultivation. So they have to feed them with organic rich compound or wastewater. It can grow on the wastewater. Okay. So some of the products you can make from these algae. Look at, you can also use it for biofuel, make hydrogen. You can make starch. From the starch you can make ethanol. You can take algal cell and make biomethane. Use the whole algae, put them in an anaerobic digester. It will digest them and make methane out of that. You can make biodiesel from the lipids from algae. And some of those high value compounds include your nutraceutical, pharmaceutical product. This rich in protein, 20 to 40%. We have this omega-3 fatty acid, the DHA I talked about, and then the red pigment carotenoid called bioactin. So these are various product commercially value to make from this algae. Very, very common in Japan, South Korea, in their what do you call that, cosmetic industries. Okay. So algal growth is quite rapid because it double really fast. The generation time, every few hours they multiply in microalgae. They're also mostly phototrophic. Some of them are heterotrophic. The difference is phototroph needs light. Heterotroph can use organic carbon. Okay? Phototrophs use light as energy source. That we went through that reaction. Heterotroph use organic carbon. So like bacteria does the fermentation or break down organic compounds. Same thing, algae can do that. Take organic carbon from wastewater and combine with the oxygen and make all the cofactor nutrient that it needs, like nitrogen, and you make biomass, CO2 and water, exactly like bacterial breakdown of organic compounds. Autotrophic growth needs carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, iron, all those things algae gets in nature. If you want to grow them, you need to provide them. Okay. Algae are about 50% carbon, 10% nitrogen, 2% phosphorus, we break it down. And then you have other algae make these pigments and oils, all that stuff they make them. Under nutrient limiting condition, especially when you low nitrogen, the growth is reduced significantly and it trigger accumulation of lipids, like I talked about yesterday in the bacteria. So algae generally prefer neutral to alkaline pH. They don't like acidic pH. Okay, so if you want to make some lipids, you need to grow algae under nutrient limiting conditions, especially nitrogen limiting conditions. They accumulate a lot of lipids. So let's look at some algal kinetics. If you want to see growth rate, how algae grow, what do you need? The kinetics look like this. The mu is your specific growth rate of algae, this mu. So algae needs sunlight, temperature, substrate concentration. That's what that F that goes into the system. You grow algae in a big pond system, so you have to make sure there's enough light in there, right temperature there. If it is all the nutrient that needs, then you'll have an output of algae. It could be depending on what you want. The whole algae cultivation, you can cultivate them, or you can get the pigment out or lipids out. Okay, so the main thing is if it is algae that needs light, you need to provide light and enough nutrient and right temperature. So steps in algal production, basically you need site selection, make sure you have a lot of light, open light source. For growth of algae, you need to, if you grow them in a bioreactor, you need to optimize the conditions, provide right nutrient, and make sure it's mass transfer is happening between the cell, getting the nutrient going in and out without any spending energy. Harvesting, after that, when you grow them, then you process the harvest. You get dewatering, thickening, drying process. Then you do the downstream processing of extracting the oil out, get the biodiesel production if you want to do biodiesel from the lipids. So that's steps in algal cultivation that shows in schematics here. Site selection where you want to grow algae, also make sure the right species is grown, and get your algae grown with the right condition, all the things that they need, light all the way, nutrient, plenty of carbon dioxide, they're right to fix the carbon dioxide. Harvest them, biomass processing, oil extraction, biodiesel production in this case, if you want to use it for biodiesel. Site selection is important, so make sure the site where you're growing algae has a lot of sunlight. It's mostly tropical and subtropical climate, and the US, these are the places they grow a lot of algae. Those are Hawaii, California, Arizona, New Mexico, Texas, and Florida. The moderate to high temperature year round, temperature is good. Adequate land availability for open pond system, lot of CO2 available in close proximity from factory. We can pipe it CO2 into the pond. Availability of water and nutrients at lower cost, and manpower is a reasonable rate, the labor cost. Okay. So this is the map of US. You can see the light intensity of the how much solar light comes into the different state. That's the light intensity. So these are the state with a lot of light. That's color right there. This is where you have a lot of algal cultivations. Okay. So then culture selection, which algae you want to grow? You want to select the photosynthetic algae that require culture vessel, light, carbon dioxide, that input into the system, and micronutrient. Some of them need cofactors. Then the right temperature, 15 to 30 degree, which is a wide range for different species for optimal growth. Then you have microalgae biomass is represented by this formula that can be used to calculate the nutrient requirement, how much they need carbon, oxygen, hydrogen, nitrogen, and phosphorus. You can calculate. All algae. Yeah. Same calculation based on the range, they put that range. Okay. For marine algae, seawater supplemented with commercial nitrate phosphate fertilizer. So you need to have salt water for marine algae if you're cultivating them close to coastal areas, and other micronutrients are used for marine algae. Okay? So here's a list of algae and the product. All the algae commercially you can produce, and I highlighted some of them. The lipid content, lipid production milligram per liter per day, this algae can make. Volumetric productivity of biomass, gram per liter per day, how much they grow, and then you have aerial productivity. Okay, so you can see all these data points, and the one that we really like them are these algae here that highlighted. Okay, that has different lot of lipid production, lot of value added product you can make out of that. So it's too small for you to read. When you get your PowerPoint slides later on, you can make use of it if you are interested in algae technology. So that information is really useful. Okay. So how they when you grow algae, they form exactly like bacterial growth curve. They go through the stage. They call that lag phase. When you get your some amount of algae put on a pond, there are not a lot of algae there. That's lag phase, and they started to multiply. That is called logarithmic phase because the doubling time is short. They double every few minutes. Some of them are. And then they use stationary phase, and then when nutrient run out, they started to die. They call it death phase. Okay, so those are your different stages of algal growth when you are growing them in a controlled conditions. Okay, of course, in nature it doesn't follow this phase. Okay, because there is plenty of opportunity for them to grow continuously or go dormant depending on availability of nutrient. But when you are growing them in a controlled setup, because of what is inputting into the system, this is what it follows. At the beginning, there's a lot of nutrient there. It's going to grow, really fast. When slowly nutrient going to run out, it plateaus out, and then it goes down. Okay. So then what you do, you harvest them, then start another batch of algae. Okay. So that's how algal growth look like. Algal production, some pictures here. The algal biomass is grown in open pond system. You can have the big pond with solar light out there. Could be 0.3 meter deep or about 1 feet deep when you have this pond. Then they make sure there's mass transfer of all the nutrient going into this bag. The algal cultivation like carbon dioxide, oxygen, and wind action, all that. We need to see the right site selection. Then sometimes the people use paddle wheels to aerate and mix the algae. And also you can use raceway pond system. The raceway is like a raceway system that flows, and that flow of the water mix the system and aerate the system, and so that's called raceway system. So I'll show you some picture of them, how the raceway system look like. So you grow algae in this raceway pond. Okay, so the water moves, in different direction, and it looks like a raceway. It's a closed loop recirculation channel for mass culture. The mixing and recirculation happens, then you can use paddle wheel to do that. So you can see the example of the raceway in a large system, and algal harvesting you can use it behind the paddle wheel too. So here's another system here. So here's the actual cultivation of algae in the US. So this is a raceway pond look like this. So the water flows. When the water flows, it mixes everything up. So the easy mass transfer happens in the algae. Nutrient move in and out without algae spending any energy. That's what raceway does. The largest facility is at 440,000 cubic meter, owned by that company called Earthrise in Hawaii. And it produces cyanobacterial biomass for food industry, for purpose of food production. Advantage is low capital cost and a low operation cost. So once you install this raceway system, operation is simple. So it's minimum input of moving the water. Okay. The one drawback is it's an open system, so water get evaporated. Okay, water evaporation, you get water loss, and then you have contamination of microbes. So sometimes, most of the time algae is okay with contamination. A little bit of contamination is okay, but when you don't take care of it with the constant mixing, then you have other contamination happen. Then you have to have separation problem. Okay, and that leads to less productivity and then when you have this in a large scale, like here, 440,000 cubic meter, that needs large land area. So you need to find a place that doesn't cost much, and it's a very expensive investment. Okay. Here's an example of another site in Hawaii called Cyanotech at Kona Island in Hawaii. And you can see the reddish pond that they're producing this algae called Hematococcus that make this astaxanthin product. Okay, and then you can see that reddish pond right here, right? The red color, that's basically algae that gives that color. Okay, then the other ponds are spirulina. You can see the green color. Okay, so all that are different colors. This is in mass production in Hawaii. And if you're using a bioreactor in a closed control system, that's how it looks like. So you need to provide light, and you have a photobioreactor system. It's a closed system, as a compact system with full control of environmental conditions. You manage everything. You don't have contamination problem because you control everything. So you can grow single culture algae. There's no problem of contamination. You allow high cell density when you use photobioreactor. Okay? In a large scale photobioreactor, that's how it looks like. You don't use the sunlight you don't use the light inside the building, it's outside. The sunlight is used as a light source. So when you have in closed system, water loss is minimum, so water evaporation is not happening. Production cost is high because you need to buy this system. It's going to 1 order magnitude higher than your pond system or raceway system. So overheating and biofouling could happen in a closed system like this. So there is some advantage and disadvantage, and you need to choose the right system for what kind of algae you want to grow. Here's another plant that produces Hematococcus plant by the company Algal in Israel. And again, the product they make is the astaxanthin and other microalgae derived products for nutrachemicals and cosmeceutical industry. Whatever pigment come out of this algae, they purify it. These are really, really high value product, very expensive product. Okay, so very profitable. Okay, because of these pigments. So we can also use algae for bioremediation, because if you have a wastewater treatment, one of the input cost is aeration. So to aerate the pond system for aerobic bacteria, because oxygen is your limiting factor. When you have a lot of carbon, the aerobic bacteria are going to consume the oxygen fast. Then you need to put more oxygen into the pond, right? That is an added cost to your treatment system. You have to electricity use pumping oxygen into the big pond system. But if you grow algae in the pond, in the wastewater treatment, algae does this oxygen production for you. You don't have to supply oxygen to the wastewater treatment system. Here is an example. So algae is grown in the pond. Algae during photosynthesis releases the oxygen. This oxygen is consumed by the bacteria. And the bacteria break down the organic carbon that you want to get rid of, reducing your BOD, COD in the wastewater. So this is greatly used in the wastewater treatment system as an oxygen supplier to bacteria, because during photosynthesis, these algae produce oxygen that could be used by aerobic bacteria to break down organic carbon. Okay. So there's an example of here algal grown wastewater treatment system, so you don't have to pump oxygen into this process. Then you can do environmental remediation with algae. Microalgae are much more effective than terrestrial plant in capturing carbon, because we are worried about carbon capturing. Algae are excellent because they can take the carbon dioxide out of the atmosphere. So when you're putting too much carbon dioxide, you want to take it out, grow a lot of algae. So marine algae are really your carbon sink. They take the carbon down from carbon dioxide. They're very efficient. The estimate is 10 to 50 times higher than a plant system. They capture more carbon. Okay. So harvested microalgae can be then turned into biofuels if you're doing this in a closed system. You're not only growing algae for your product, but you're also doing a favor of capturing carbon dioxide from the atmosphere. Okay. So carbon capture is good. Carbon is fixed, and then the algae make your product, and then you can use the algae to make biofuel or anything else that you want to do that. The negative impact of algae is when you pollute your water system with too much nitrogen and phosphorus, algal will bloom. We call that algal bloom, and that is an environmental problem, right? It happens every year in the U.S. in coastal area in Florida. Unwanted algal bloom, we call that red tide toxin. The red algae produce some toxin. When you go swim in the water, you get sick, all right? So that happens. Here is the red tide, and you can see the green tide in China. So that's because of too much nitrogen phosphorus going into the coastal water. Algal bloom happens and really produce some toxic chemicals. Okay, and also cause hypoxia. That because oxygen sucked up, the dissolved oxygen is low, all the fish going to die. It's called fish kill happens. Okay. So that's a overall view of a lot of algae I talked about. Pigment producers, you can use algae for different things, biofuel, pharmaceutical, and also cosmetic industries. And now I'm going to concentrate on my research with algae. What we do is to use algae to remove emerging contaminants in the environment, especially antibiotics. So we produce a lot of antibiotics, a lot of antibiotics end up in the waterways, and the efficient way to control is use the algal system. Okay, I'm going to show some of the research in my lab. Okay. So the best way to see the algae in your natural system is wetland. If you have a natural wetland, you will find this algae floating everywhere. Okay, so this wetland is close to my campus. We do research in this wetland. So what's the main input of water to this wetland is the sewage treatment. After sewage is treated from the city of Thibodeau where I live, the treated wastewater is released into this wetland. The treated wastewater still has residual carbon, nitrogen, phosphorus. That promotes the growth of algae. Okay? The treated wastewater also contains a lot of antibiotics. Okay? And the antibiotics are not taken out by sewage treatment system. Sewage treatment system is not designed to remove antibiotics. They are designed to remove carbon, nitrogen, phosphorus. Okay. So we pollute the environment with we call emerging contaminants that include antibiotic, nanoparticle, microplastic, pharmaceutical, personal care product, all the makeup stuff we apply. Everything is not degraded in your sewage treatment plant, everything going into the water after the sewage was treated and released. Okay, it all ends up in the natural system like this one, and I'm going to talk about one particular aspect of how this algae is helping us to take down antibiotics. So a little bit of history about antibiotics. So if you look at antibiotic discovery, the first antibiotic was discovered accidentally in the 1928 by Alexander Fleming. Was originally intended to discovered, accidentally discovered penicillin. because of contamination of the fungus Penicillium in his petri plate. And that production of antibiotics, helped save a lot of life and infectious disease control. So you can see that from that time onwards, a boom in our production of antibiotics. And this is the golden age of antibiotics. Okay. After 1960, very few antibiotics were discovered. Right? People, because the pharmaceutical industry, moved on. This is not a money-making scheme. They moved on to making drugs that cause lifestyle disease like obesity and diabetes. That's where the money is, right? So they're not spending money to produce new antibiotics. So antibiotic discovery declined, and now we have no new antibiotics out in the market. We are using the same old antibiotics to treat. Very, 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 very few, one or two coming out every 5 years. So that's the problem of overuse of antibiotics, and we have problem treating some diseases. Okay, so antibiotics are used in a lot of things for treating infectious disease, and also for your, as an anti-infection in your makeup industries and cattle industries and farming industry. These are all places we use antibiotic. And just to look at, there are different classes of antibiotics, just to show you based on how the antibiotic work. We have sulfonamide, quinolone, beta-lactam, tetracycline, macrolide, aminoglycoside. They all have different mechanism to kill bacteria. Okay, so that's there are more than 265 commercially available antibiotics. They all fall under these different family of antibiotic. This is how the mechanism of how antibiotics work. Sometimes they stop protein synthesis in bacteria, sometimes they stop DNA synthesis, sometimes it stops production of cell wall synthesis. That's how antibiotics are classified, the mechanism. Okay. So let's look at the problem of antibiotics. Okay. In the US, this is the US statistics, 270 million prescription per year, for 300 million people, which is 2.8 million antibiotic, result in 2.8 million antibiotic resistant infection. These are people have bacterial infection, they cannot get treated with a normal antibiotic, and that results in 35,000 deaths every year in the US because of antibiotic resistant infection. Okay. So the problem with antibiotic, when we take antibiotic, 30 to 90% of the antibiotic you take is not digested in your system. It just comes out in your urine and fecal matter that end up in the sewage and released into the environment. So only 10% or maybe maximum 30%, it can be absorbed in your body for your treatment. Rest of them comes out as active compounds. Okay, that's the problem of antibiotic into the environment. So how antibiotic release in the environment going to cause antibiotic resistant bacteria? That's a mechanism here. So there are three phenomena that can do: transformation, transduction, conjugation. Transformation is when bacteria die, this bacteria is antibiotic resistant bacteria, releases the gene. The bacteria that are sensitive to antibiotic can take the gene and incorporate into it and become antibiotic resistant. That is called transformation. And transduction, the viruses can accidentally transfer gene from one bacteria to another bacteria. That is called transduction. And conjugation is two live bacteria exchange the genes. And these are the various ways bacteria exchange genes, and this is called horizontal gene transfer. Bacteria is the only organism can transfer gene horizontally between two adult cells. Okay? The vertical gene transfer is when we reproduce, we pass on the gene. So that is, once the bacteria get the new gene, every time they multiply, the antibiotic resistance spreads. Right? This is vertical gene transfer, this is horizontal gene transfer. That's what's happening in the environment. Okay? When we have few antibiotic-resistant bacteria, they transfer the genes to bacteria that are sensitive to antibiotic that become resistant. Every time that bacteria reproduces, you get a lot of antibiotic-resistant bacteria. So these are the various mechanisms of antibiotic resistance. Bacteria develop mechanism to make enzyme and make antibiotic inactive, and they change the ribosome structure so the antibiotic cannot go bind, so antibiotic become useless. And then it has a proton pump. It can pump the antibiotic outside the cell. When antibiotic get into the bacteria, it quickly comes out. So those are various way bacteria develop mechanism to resistant to antibiotic. So in another way, the antibiotic resistance spread is when you take too much antibiotic and when you don't need antibiotic and you're taking antibiotic, and this is what happens. So in your body, in your microbiome, all the good bacteria die out. Only few antibiotic resistant bacteria will survive, and then they have no competition, so they multiply really fast, and you end up having too much antibiotic resistant bacteria in your guts. Okay, so if you overuse, misuse antibiotic, that's what happens in your body. Okay. So, as I said, wastewater treatment, primary objective is to remove carbon, nitrogen, phosphorus. They are not designed to take antibiotics, right? So all the antibiotics that ended up is released 100%. What comes into the sewage goes out. Nothing happened to these antibiotics. So that's how a sewage treatment plant looks like schematically. You have a primary treatment, secondary treatment. That's where the bacteria reduce your BOD, COD, and then you have tertiary treatment, disinfection, and then this is where we are letting it out. In our case, it goes to the wetland. Okay, the wetland has a lot of antibiotics in that. So that's how when you treat in the wetland, you have algae, you have bacteria, and the algae can absorb this antibiotic, and also algae can help bacteria to break down because it produces oxygen and other bacteria to degrade this antibiotic. So algae play a key role in controlling this antibiotic resistance in natural system. So this is what's happening. You have wetland, easy to operate. It filters out antibiotic, and it degrades and also entrap antibiotics. So when your wastewater comes in, here is your wetland. You have a lot of algae, a lot of plants, and it's all antibiotics entrapped here. Some of the antibiotics degraded, some of the antibiotics filtered out. So whatever the water comes out of the wetland is less antibiotic, and that's the principle behind using algae, using wetland to control the problem of emerging contaminant. Okay. So what is happening in the wetland? There is physical absorption, chemical oxidation, photodegradation, biodegradation, all the phenomena happening in one place, right? That's why it can take out this emerging contaminant. Operation wise is low cost, low operation maintenance, and limited secondary pollution, because all you're doing is let the water go through the wetland and it comes out on the other end, and the algae and bacteria and plant will take care of this. Okay. If you look at the antibiotic removal in regular conventional process, UV light can reduce your antibiotic, membrane bioreactor can take your antibiotic, and the third one is your constructed wetland, which is the cheapest one. Okay. So goal is to look at whether our wetland can do this job with algal production. So that's our goal of our project. That's our wetland. You can look at algae right all over the surface of the water. We looked at the indicator organism like fecal coliform, total coliform bacteria. We looked at two species, 1 gram positive, 1 gram negative bacteria that are resistant to antibiotics, whether it is reduced in this system. And we looked at what happened, the water goes in, what happened, the water comes out of the wetland. We monitor in winter and summer months. Okay. So a little bit of research is just to orient yourself. That's a sewage treatment plant, and this is the wetland right there. The water comes for 2 miles piped underground into this wetland. And this is your sewage treatment plant, and the water from here is piped. These are the pipes into the wetland. Okay. So that's how it looks like, where the water is coming out of the sewage. This is water from the sewage plant. After it's treated, is series of pipes all along the wetland goes into the wetland. And you can see plenty of algae, all kinds of species of algae in there, right? That's an important point to stress here. So we took sample in different sites. So let me quickly go through this. We luckily had a control site. The control site is not receiving water from sewage plant. It's totally impounded separately. That has less algae, no much contamination. We can compare the performance of our system. So that's the control site. The control site, you don't see algae, you see plant, right? So we collect sample every month. My grad student went there and collected sample, brought to the lab. These are our different sites. Control site, the water comes into the wetland, midpoint, water goes out into the river. Okay. So we did real-time sampling of dissolved oxygen, temperature, salinity, all that right at the site. And then we collect this, that's my student collecting sample, bring it to the lab. We did nutrient analysis, carbon analysis, nitrate, phosphate, ammonia, sulfate, everything she did. Then quick results, and we did total coliform, and then we identified antibiotic resistance using this Kirby-Bauer assay, put different antibiotic and put them, bacteria come out of the wetland and see whether they're resistant or sensitive. If it is resistant, they grow close to the antibiotic. If it is sensitive, they make this zone of inhibition. So that's how we see whether bacteria are resistant or sensitive. So we have a good record collection, record keeping, just to show you our really good. And then we isolated these two indicator organisms, 1 gram positive, 1 gram negative bacteria, and tested their antibiotic resistance. And this is a quick result. You can see 4 sites, and there is no major difference in dissolved oxygen, temperature, pH. The only difference is pH was a difference. The pH is in the site, in the midpoint, and the outpoint is acidic, and compared to control, and the endpoint is more than neutral. So this is a lot of acid production in the pond because of multiple organisms digesting the carbon. And nutrient analysis, to orient yourself, you can see site 1, 2, and 3, and 4. Site 1 is a control, site 2 is where the wastewater coming in. This is midpoint, this is where it is going out. So you can see all of the cases, most of the cases is high nutrient coming in, and then what is going out is comparable to our control. So all the nutrients are removed in the wetland, whatever excess nutrient coming from the sewage plant, taken by bacteria and algae is removing the nutrient. Okay? And just to compare the seasonal variation in the summer month and winter month, on all 4 sites, the same trend. The input site has more concentration than the output site. The output site concentration equal to control site, which is exactly what we thought is going to happen, and that's what the result looked like. Okay? In every parameter we measured, whether it is nitrate, sulfate, phosphate, ammonia, all that stuff. Just to show you color, how each sample look like after you do the test. Okay? And then we measured the bacteria we call total coliform, fecal coliform indicator organisms. You can see the control site, inside, mid-site, outside. You can see the inside has a lot of this fecal coliform, outside has no fecal coliform compared to control, same data. So what's happening in these algae is filtering them out, and that really good for the system, natural system. And then we did the bacterial diversity analysis, what kind of bacteria are there, and that's a list of the bacterial phylum level and different site. control in and out and middle side. It is compared with what is in the water, what kind of bacteria is present in the system. Okay, and then we look at the different genus, did some molecular work, and you can see a lot of diversity of organisms in the water, and some of them are dominant. The color coded, you can see the green is dominant, and you can look at family level. These are more data to show what is in the water. And you can see these are the dominant one, the one that is highlighted. And then you can look at the antibiotic concentration. Okay, we look at amoxicillin, erythromycin, sulfonamide, tetracycline, bacitracin, penicillin, and all that antibiotic in different site, and you can see the difference in the concentration. So you can see inside, this control side, this is inside, the outside. So the lot of antibiotic coming in, less antibiotic going out. The wetland is removing these antibiotics that's going into the system, mostly by algae and also bacteria doing this work. Okay, so, and just to give you information about antibiotic resistant, Kirby-Bauer chart, we measure the zone, that's the zone in millimeter for different antibiotic. All the antibiotics are listed there. And that's how Kirby-Bauer look like when you test for antibiotic resistance. You can see these plates that has more resistant in site 1 compared to site 4. Site 4 has less resistance. Okay, that's good. They're taking out antibiotic resistant bacteria as well. Okay, so that means this one bacteria here is resistant. Grow close to the disc. More zone means sensitive. This antibiotic is killing the bacteria. This antibiotic is not killing the bacteria. All right, more pictures to show what's happening in each other site. And the take-home point here is we are reducing the resistance to antibiotic in the sewage treated wetland. Wetland is doing a job of tetracycline, sulfonamide resistant bacteria went down. Okay, and just to list all the antibiotic and all the resistant in species wise, E. coli, Staph aureus, total resistance, as show you here. Okay. Again, the same point in percentage wise, how many percent of resistance we are seeing here. You can see 50 to 75% of resistant bacteria is removed because of algal action and bacterial action in the wetland. And see the different species, E. coli and Staph aureus. Okay. As a total resistance, we put both data together, that much resistance is removed. So then one more thing we did was we extracted the genes in the water, in the wetland, and took out the DNA in the water and looked for antibiotic resistant genes, whether there is antibiotic resistant genes floating in the water. And you see there is primers for different antibiotic genes. This is listed here for different antibiotic. These are the genes we looked at for tetracycline resistant, sulfonamide resistant, erythromycin resistant. These are the gene code primer we use, and that's the length of the gene. We looked at base pair, and then you can see these genes are present in the inside. So the antibiotic resistant genes are present in the site. You can see that? Compared to control. This is your control, which is the positive control, to make sure that what we are doing is right. And these are the sites that shows the antibiotic resistant genes. So there are not only antibiotic there, not only antibiotic resistant bacteria there is also free floating genes. Okay, these antibiotic resistant genes can survive for 96 hours before it fall apart. During this time, when a bacteria that is sensitive to antibiotic come in contact with the gene, as I showed you at the beginning, it can transfer into antibiotic resistant genes. Okay? So this wetland is taking out some of these genes as well. So So multi-drug resistant bacteria are found in the input site more than output site. Okay? So multi-drug resistant bacteria are bacteria that has resistant at least 3 or more antibiotics. MDR we call that. Okay? So overall, we talked about biofuel and I went into wastewater treatment. Just to show you, the algae has a different use. Right? It's a very versatile organism. Your biotechnology of algae use could be for fuel production, pharmaceutical, cosmeceutical industry, or wastewater treatment. And it is cheaper, easy to manage if your wetland using the wetland as a treatment choice. Algae grow really, really fast, and lots of algae in the wetland. Okay. So just conclude my student did the work, finished her graduate work, and we confirmed antibiotics and resistant reduction, antibiotic-resistant gene reduction in our system. And I want to give credit to some algal technology slides to my colleague Samir Kanal. All those industry, he took the picture, and I'm sharing that credit slides for you from him. Okay, thank you, and I'll take some questions. Yeah. Yeah, that.
Sir, thank you for your wonderful insights on algal and algal-based treatment systems.
Mm-hmm.
Sir, particularly about antimicrobial resistance.
Yeah.
I was wondering about any mechanism you found when you moved from site 1 to site 4.
The removal of antibiotic?
Yeah. So
yeah.
Any specific mechanism you draw how this removal mechanism is occurring from site 1 to site 4?
Yeah, it's a combination of everything, but the majority of them is algal. Okay, but as I said at the beginning, antibiotic degradation is adsorption, filtration, photodegradation, biodegradation, and algae also take up and absorb, and so water comes out with less antibiotic. So that plays a role. When there is less antibiotic, there is less antibiotic resistant bacteria multiplying, right? So antibiotic is bacteria develop resistance when they are exposed to antibiotic. So when there are less antibiotic, there are less chance of them developing resistance. So it's like a concert of a lot of different mechanism work there.
Sir, it can be figured out like controlling the antibiotics right at the source. Right at the source. As you are saying that 30 to 90% of the antibiotics is going wasted into the wastewater.
Yeah, that's hard because you and I are taking antibiotic, right? That's our source. You cannot control. If you're sick, you take antibiotic, but you don't want to misuse it. You don't want to overuse it. So it's a public education. You need to talk to doctors, talk to patient. Hey, don't dump the antibiotic in the water. Properly dispose them, and if you take them, don't overuse the antibiotics. And everything that you take in terms of antibiotic and medicine, only 10 to 30% is absorbed. Rest is coming out and end up in.
So it's ultimately become the question of public education.
Yes.
And how the doctor.
You have to start in your kindergarten level. It's a generational thing, right? So people won't change overnight. So we need to educate the people, hey, don't do this. And because it's going to be a big problem because we are using newer chemicals, and PFAS is another one, which is we call forever chemicals. And I have a project on that. So it's too many chemicals that we call emerging contaminants and microplastic and all that stuff. Yeah, you're right. Source reduction is the way to go. Source reduction is human, so we have to educate ourselves. Yeah.
Thank you.
Yes, antibiotics uptake in the plant and trapped in them, and there are some antibiotics in the roots, and some of them are filtered out in the sediments itself. So that's another story. The water is clean. The wetland has some
accumulation of antibiotics will
in
be
there, but what we are doing is we are trapping them. We are keeping them in one place instead of spreading further and further in your downstream water body. So there is degradation, of course. Over long time, when this antibiotic are in the root system, it eventually get degraded because of not only plant enzyme but also rhizosporic bacteria. The bacteria live close to the root, also degrade these antibiotics over time. It takes long time. Yeah.
like similar to microbial population, that antibiotic resistant gene transfer, like similar thing can happen to the plants also, whether this transformation can happen.
Plants, not a problem at all. The antibiotics really effective only about bacteria. Plant has no problem with antibiotics. Yeah. 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 Yeah.
So you said that the algae can be microalgae can be used for the aeration purposes in wastewater treatment.
Yes.
So when the microalgae gets exposed to a different microenvironment other than its comfort state, it goes under dormant stage or death stage, right, sir? So how can it undergo aeration?
That death stage happens in the control system. When you grow microalgae in a lab, in a controlled bioreactor, it goes through lag, log, stationary death phase. When you grow in the algal pond, it's an open system, it doesn't follow that. Because there is a continuous input of carbon coming in, right? And continuous input of sunlight. So it doesn't follow that pattern. So it won't die. It continuously multiply. As long as there is no limit, there is no limiting factor. Plenty of nutrient, plenty of sunlight, algae is happy. They don't die. They continuously produce oxygen, like a continuous aeration in your system. Yeah.
Thank you, sir.
Yeah. Yeah.
In the constructed wetland system,
Yeah.
if algae is completely grown and it is completely filled over, there is a possibility of getting an anaerobic condition, right, sir?
You don't want too much algae in a small pond, but this one is not it's a natural system. The water flows out. In constructed wetland, if you don't have water outflow, you will have over algal production. Okay, so that will lead to problem. Yes, yeah. But the system, there should be a continuous flow of water. The algae in the middle doing the job,
Okay.
and then it's a slow moving water. And the end it gets out. The water that comes out is pure, and it takes out all the nutrients, all the contaminants. Yeah.
If it is a still condition, it may develop an anaerobic.
Exactly. Yeah. If there is no flow, then you'll have algal accumulation. Yeah. Yeah. Yeah.
Thank you.
You want to look at the Zoom questions? Okay. Yeah. Yeah. Antibiotic doesn't work in algae. Antibiotic works only in bacteria. Algae doesn't get killed by antibiotic. All your antibiotics are only for bacterial infection. And a viral infection also, it doesn't work on virus also, right? Only very few viruses. Antibiotics only for bacterial and fungal infection. Algae don't care, plant don't care. Antibiotics have no problem with those. Only it's effective on bacterial and fungi. Yeah. Any questions from the Zoom people? Yeah, all right. What may be the minimum capacity required for raceway pond? The question is based on how much you want to produce. Okay? You can make your raceway as small, as big. Your investment depend on investment, because the raceway, at the beginning you have to invest. So it all depends on what you want to produce, whether it is a pharmaceutical, nutraceutical algae you want to produce. You want to make a huge investment. That is good. Yeah, that answer is not one answer fits for all, right? It's a tailor-made, custom-made question, that custom-made answer need to be answered. So you need to know what is the purpose of your algae, whether it's for biofuel, whether it's for pharmaceutical purpose, and then how much you're going to produce. So it's all based on that. You have to answer the question. So I can say that, oh, this is the system you want, this size. It doesn't, one size doesn't fit. Okay. Okay. Any other questions?
Any other question from the online candidate? You can unmute and you can answer the question.
If nobody raised any question, hands, if there is a hand raised on there, I can. Okay. All right. It looks okay. 12:30. Okay.
Thank you, sir.
Okay, thank you all the participants. So now we have break for lunch.